Our lab's focused on several different lung diseases. They're actually quite rare lung diseases. One is cystic fibrosis, you've probably heard of that. The other is a series of diseases that are grouped together as called primary ciliary dyskinesias. Both these diseases can strike in the newborn. They're both genetic lung diseases, so they're caused by mutations in the genome that cause defects and something that leads to defective lung function. And we're using human stem cells to study these diseases because they're actually there's a big lack of animal models that really recapitulate how the disease forms in humans. So even though we can find something that may treat a mouse, quite often that does not then translate into the humans, and we're looking at completely different mechanisms that are driving disease pathogenesis in humans. What we're doing is actually we're going directly to the patients and getting cells from the patients. So we're either obtaining blood samples or getting a skin biopsy. We're able to take these cells to the lab, and we're able to then convert them into what we call a pluripotent stem cell. So this is a de-differentiation or a reprogramming of the cells from being a mature cell to being a very immature cell type that in a dish we can grow them indefinitely in the lab, and we can also push them to become any cell that we want if we give them the right signals to become those cells. So in our lab, we're taking these pluripotent stem cells and pushing them to become airway epithelial cells, so the cells that line the airways. And we have these cells in a lab and we have them from patients. What we can do is we can actually go in and genetically correct these patients, the mutations that cause the disease. So now in the lab, we have a great model system where we have the diseased patient cells and the gene corrected, so we can study specifically what that mutation is doing to our cells. And not only can we do this in epithelial cells, we can look at it at stages of lung development as we push these cells from being stem cells to being mature lung cells. And we hope to be able to use these cells to be able to model the disease so we can try and generate screening tools. So we can take a precision medicine approach to treating these diseases. And we also hope to use the stem cells to look at how they can be used to repair or regenerate an injured lung epithelium. Every person has their own genetic makeup, and everybody who has a disease has their mutation on top of their own individual genetic makeup. So while the disease-causing mutation can have a certain effect on your cells, the genetic makeup of an individual can actually affect how a person responds to a certain therapeutic approach. So by taking individual patient cells and making model systems, we can then put these into a high-throughput screening tool where you can look at the effect of drugs on that patient's specific cells and their specific genetic makeup to find a drug that actually is successful in treating that individual. And one treatment may not be equally as effective on all patients.